Hey there, BioBuds. We are going to wrap up Unit 3, Topics 6 and 7 by talking about what happens to respiration when you don't have oxygen. So we've already talked about in Part 1 going through glycolysis and then pyruvate oxidation, the citric acid cycle, and ultimately oxidative phosphorylation to make all of my wonderful ATP in the mitochondria. But what happens if I don't have oxygen after glycolysis? So remember, in cellular respiration, oxygen is the final electron acceptor. So it allows electrons to move down the electron transport chain. So what happens, or what do organisms do to produce ATP if there is no oxygen? Well, there's two options. There's anaerobic respiration and fermentation. So remember, the prefix a or an means without. Aero means air, which in this case is referring to oxygen. So we'll start off with anaerobic respiration. So this is whenever I'm generating ATP using an electron transport chain that works without oxygen. So this is specific to prokaryotic organisms. Eukaryotic organisms require oxygen as their electron acceptor. So this is bacteria only here. And what happens is that they have a different final electron acceptor. They actually use sulfates or nitrates instead. So since oxygen is the normal final electron acceptor, these organisms have adapted to use something else instead of the oxygen. My other option is fermentation. So this is generating ATP without using an electron transport chain. So this is just going to be an extension of glycolysis. And my whole point here is I have to reduce NAD to get rid of those electrons. And this is going to occur in the cytosol. So no oxygen, no mitochondria. Okay, we only move into the mitochondria if we have oxygen. There's going to be two types of fermentation that we can talk about here: alcoholic fermentation and lactic acid fermentation. So let's take a look at alcoholic fermentation. This is whenever pyruvate that is created by glycolysis is going to be converted into ethanol instead, which is a type of alcohol. So examples of this are bacteria and yeast. This is why yeast is used in most productions of alcoholic beverages, because basically ethanol is like yeast pea, right? It's a waste product of the yeast doing their fermentation, their type of respiration. So glycolysis still happens where I'm taking glucose and I break that down into 2-pyruvate. And during this whole process, I've made 2 ATP. That still happens. So this part is still the same as cellular respiration. Okay? It's just glycolysis still. However, now I have these 2-pyruvate. I've created these NADHs, right? They have their hydrogens. The dump trucks are full in the back of electrons. So I have to reduce them in order to use them again in glycolysis. So how they fix this is they turn the pyruvate into acetaldehyde and then ultimately into ethanol. So what that does is whenever I'm taking the acetaldehyde and converting it into ethanol, it's going to reduce my NADHs back to NAD pluses so that they can go back and accept electrons during glycolysis. And ultimately, this is just going to produce carbon dioxide as a waste product. So this is why when you are baking bread and doing yeast or using yeast, you're going to bake out the ethanol in your yeast bread dough. So your bread's not alcoholic. However, why does it rise? Because of basically like yeast farts here, right? The carbon dioxide are producing these bubbles, which cause the bread itself to rise. Looking on to lactic acid fermentation. So once again, I still have glycolysis happening, just like cellular respiration. I have my molecule of glucose. It gets broken down into 2-pyruvate. I have 2 net ATP that are made in this situation, but I also have 2 NADHs that I have to reduce somehow. So instead of using pyruvate and creating ethanol, I'm going to use my pyruvate and create lactate. So that whole process of taking my pyruvate and turning it into lactate is going to take my NADH back to NAD+. That way it can go back to glycolysis and assist with that reaction. Because remember, glycolysis is how they're getting their ATP here. So I have to recycle the things that are needed for glycolysis, and in this situation, that's NAD+. Why lactic acid fermentation is important to you is because you do it. And you might get a question about muscle cells. So when your muscle cells are going to run out of oxygen, they can go through lactic acid fermentation to help produce some more ATP for your muscles. Whenever you have a buildup of lactic acid, that's what's going to cause that burning sensation whenever you're performing kind of strenuous exercise. So know that lactic acid fermentation happens in your muscle cells, and the buildup of lactic acid or lactate can cause a burning sensation. Well, what does your body do with the lactic acid? Like, just leave it there? No, it does not. So your muscles will produce that lactate, which will then go into your blood and is broken back down into glucose in the liver, which totally makes sense because we know that livers are detox organs. So anytime where you're like ingesting anything that's really not that great for you, or your body's making something that's not super good for you, your liver is responsible for breaking that down. And remember, shout out to Unit 2 here. Liver cells have lots of smooth endoplasmic reticulum which help with this process. But anyway, when lactate is in the blood, it lowers your pH. And that means if lactate builds up and it's unable to be broken down, it can lead to something called lactic acidosis, which is when you have excessively low blood pH. This is not good, okay? This can cause a lot of complications and even death. But the good thing is, this is rare. Usually it's like because you're taking medication that inhibits your liver from functioning quite correctly. So don't freak out if you're working out and you get burning muscles. It doesn't mean that your body's going to shut down due to lactic acidosis, okay? So you're working out really hard. Your muscles run out of oxygen. They start doing lactic acid fermentation to make a little bit more ATP for you. You have that buildup of lactic acid or lactate, and that causes this burning sensation. That lactate gets moved into your blood, goes to your liver for your body to break it back down. The end. So looking at respiration and my different options, glycolysis is going to happen no matter what. Okay, it's always going to happen in the cytosol. If I have oxygen, then I am going to, one, create more ATP. So that is the more preferable method. Oxygen is important because it gives me more ATP. So I would go through pyruvate oxidation, citric acid cycle, and then oxidative phosphorylation, meaning my combined efforts of the electron transport chain and chemiosmosis. So that makes 30 to 32 ATP. Much, much higher than if I only go through glycolysis and then some form of fermentation. But if I have no oxygen, I'm going to go through fermentation. My two options are either lactic acid or alcoholic fermentation, and both only create 2 molecules of ATP, those original 2 molecules that were made during glycolysis, and that's it. So it's not preferable, but if that's all that you can do, or if that's all that you got at the time, better some than none. So go ahead and look at the next 2 FRQs. Pause this video and do them on your own, and then come back when you're ready to go over them. Alright, this first practice FRQ is talking about fish. The catch and release method of fishing was once thought to be a beneficial method for conserving the number of fish in a body of water, but it is now known to be potentially deadly for up to 72 hours after a fish has been caught. When a fish is caught and pulled out of the water, it cannot intake oxygen. The fish experiences extreme stress during this time, which triggers shimming, a condition where fish will use their muscles to vigorously shake back and forth. So think about like fish flopping on the deck, right? They're not breathing because they get their oxygen through their gills in the water. So they can't intake oxygen at this point. Identify the stage of cellular respiration that is directly affected by the lack of oxygen. Well, in your head, you should be thinking, oh, oxygen is the final electron acceptor, and that's important to oxidative phosphorylation, specifically the electron transport chain. B, explain the importance of oxygen for cellular respiration. So that's where you're telling me all about the fact that oxygen is a final electron acceptor, and without it, the electron chain is going to be shut down, which means no chemiosmosis is going to happen, and there's just going to be a major lack of ATP inside of that fish. C, explain how the fish is most likely able to generate energy to shimmy in the absence of oxygen. Well, shimmying means that muscles are contracting. And I know without oxygen, muscles can still function because of lactic acid fermentation, because lactic acid fermentation is a type of fermentation, and that can occur without oxygen. In our second FRQ, examining the permeability of mitochondria revealed that the outer membrane was permeable to H+ ions. After discovering this, a researcher wanted to study the effects of pH on the activity of mitochondria. Mitochondria from liver cells were isolated and exposed to solutions at a pH of 9, 8, 7, 6, and 5. The results indicated that mitochondria were most active at a pH of 5. Explain how pH may affect the relative concentration of ions in the mitochondria. So A, I know that anything that is acidic, meaning it has a lower pH, is going to release more H+ ions. So that is leading me to this next thought of B, propose a model to explain how a low pH could increase the productivity of a mitochondria. So I would talk about the fact that an increase in my H+ ions is a proton gradient. It helps build that proton gradient, which is going to power chemiosmosis. And chemiosmosis involves that ATP synthase, the hydrogen ions moving through ATP synthase to power the formation of ATP by bonding ADP to a phosphate. So more protons, more chemiosmosis, more ATP. It's more efficient. And that wraps it up for Unit 3, guys.